If one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin supports high copy number. Before transferring the bacteria into the host cell, it will be grown in a culture plate. When the foreign DNA is introduced into the bacteria, the foreign DNA can pass through the pores or the holes and it will get incorporated there. Hello everyone, a warm welcome to today's session on chapter 11 of second PUC, that is Biotechnology, Principles and Processes. I am Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the recombinant DNA technology, wherein we studied about the different tools that are used in recombinant DNA technology, wherein we studied about the ligases enzyme, and before that the restriction endonucleases enzyme. In today's session, let us discuss about the cloning vectors. So why are cloning vectors important? So we shall look into. So talking about cloning vectors. So cloning vectors are the vectors, or they can also be called as the vehicles that are used at present, and these vectors are engineered in such a way that they help us to keep a link of the foreign DNA and the selected recombinant DNA from non-recombinants. That is, we need a recombinant DNA. So, we need to isolate the DNA strand or cut the DNA strand from the foreign DNA and from that of the vector's DNA. So, once we cut it, then we need to introduce the foreign DNA into the vector's DNA. So, for that particular purpose, we need a vector. So, that is why cloning vectors, and we need to produce multiple copies of those vectors. So, therefore, because those or multiple copies of the gene, that is the foreign DNA, we need to produce multiple copies. Now, where have we input the foreign DNA into? We have put the foreign DNA into the vector. What is that vector? That vector is the bacteria. So, how the foreign DNA was put into the vector, we have studied in the previous session. So, just have a look into that particular video session. So, now we have put that particular DNA, that is the foreign DNA, from plant A. I have taken that particular DNA, it is a foreign DNA. Why it is called a foreign DNA? Because from somewhere I am taking it and I am putting it into the vector. So, therefore, it is a foreign DNA, right? So, think of this. So, we say foreigners. Why do we use the word foreigners? Because in India, if somebody comes to India, if somebody comes from another country, that person will be a foreigner in India. Likewise, if we go to another country, we will be a foreigner to the people of that particular country, right? So, now here from where the foreign DNA is coming, it is coming from plant A into the vector, right? So, therefore, it is called a foreign DNA. Now, the foreign DNA has been put into the vector. So, that vector, it is a bacteria, and obviously bacteria will multiply. So, when they multiply, what happens? This foreign DNA will also start to multiply in the bacteria, and it will multiplying means it will produce clones of that particular foreign DNA. That is why it is called as a cloning vector. So, there are some features which actually facilitate or help the cloning into a vector. One is because the vector has a region called as origin of replication or ORI region, and it has a selectable marker region, it has cloning sites, it has vectors for cloning genes in the plants and animals. So, that is why, because all these are present in that particular vector, it is used as a cloning vector. So, what all it has? It has the ORI region, then it has the selected marker region, it has the region for the cloning sites, and it has the region for the cloning of genes and all that. So, that is why it is one of the best, bacteria are one of the best used cloning vectors. So, let us study about each of these parts of the vectors one by one. So, the first one is origin of replication, also called as the ORI region. So this particular region, origin of replication, because the DNA strand is actually introduced. So from where the DNA strand, the foreign DNA strand is inserted into the vector's DNA at the origin of replication region, and from this particular region the replica or copies of the foreign DNA are produced. That is why it has been named as origin of replication region or ORI region. So this is a sequence from where replication starts. So what is replication? In molecular basis of inheritance, we have already looked into it. So please have a look into that particular video session. So this is a sequence from where replication will start, and any piece of DNA when linked to this particular sequence can be made to replicate within the host cell. Now what we do is, we have introduced the foreign DNA into this particular region of origin of replication, and this particular vector, that is a plasmid vector, has been put into plant B, right? So therefore this vector is carrying the desired DNA from plant A into plant B. Now in the plant B, when this particular bacteria starts to multiply, in this origin of replication region, the DNA strand, the foreign DNA strand also starts to multiply. That is how the DNA gets incorporated. Many number of DNA gets incorporated into the plant B. So that is how it is done. So this sequence is also responsible for controlling how many copies of the foreign DNA has to be produced. So that is also one of the characteristic feature of origin of replication. First thing is, it helps in making the number of copies of DNA in the host organism. Second thing is, how many copies of that foreign DNA has to be produced will also be decided by the ORI region or the origin of replication region. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin supports high copy number. So that is why different types of plasmids are used based on the ORI region. So for every experiment, the same plasmid will not be used. Different types of plasmid vectors are used, because how many copies we need, based on that the plasmid vector has to be designed or it has to be selected. Next part in that is selectable marker region. So we finished studying with origin of replication region. Next is selected marker or selectable marker region. So selectable marker region, in addition to ORI, the vector requires a selectable marker. So what is the function of the selectable marker? So this selectable marker, it helps in identifying and eliminating the non-transformants. So those genes which are not capable of expressing, or the non-transformants, that will be removed. So what will be usually done is, before transferring the bacteria into the host cell, it will be grown in a culture plate. To that culture plate, in this particular region, a selectable marker will be added. What is it, selectable marker? Again, it is one strand of DNA itself. It is a DNA strand that itself that will be added here. So DNA strand which acts as a selectable marker, that will be added there, and then it will be looked into whether this selectable marker, whether there is any non-transformants are there. If there is any non-transformant, such vectors can be eliminated and only the good vectors can be used for further experimental studies. So transformation is a procedure through which a piece of DNA is introduced into the host bacterium. So that we will study in the coming slides. So here, normally the genes encoding resistance to antibiotics. So what are the different selectable markers that are used? So why is selectable marker important? To eliminate the non-transformants. So which type of selectable markers are commonly used? So the commonly used selectable markers are those genes which are resistant to antibiotics. Why are they used? We shall look into. Say for example, those genes which are resistant to ampicillin, chloramphenicol, tetracycline or kanamycin, etc. So these are the different antibiotics that are there. Those genes which are showing resistance towards those genes which encode resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline or kanamycin. All these are used as a selectable marker or a selected marker. So therefore, because why they are used is because they are the selectable markers for E. coli. Because this is the vector that we are using, the plasmid vector that we are using is from a E. coli bacteria. E. coli is one of the common bacteria that are used as a vector here. So the normal E. coli cells, they actually do not have any or they do not carry any genes that are resistant against any of these antibiotics. Because they do not carry any genes, we will be the one who is introducing the gene. We will be the one who is introducing the gene that is showing resistance to the particular antibiotic. That gene is called as a selectable marker, and this selectable marker will choose only those vectors which are having transformants, and those having the non-transformants will be eliminated. Next, moving further, we shall study about the cloning sites. So now we know about the later on, for you to understand how exactly the cloning, and before introducing into the host what all is done, to understand that you need to know parts you need to know, especially the ori region, then the selectable marker region. Knowing these two are very, very important. So now you know at least what these two are. So later on when we explain in brief, you will understand it even better. Let's talk about the cloning sites. So cloning sites, what are cloning sites? Those regions in which where we have to link the DNA. Which DNA? We have to link the foreign DNA. Because if we link the foreign DNA in the cloning site, then only they will produce clones or copies of it, right? We cannot link it somewhere in the vector DNA. That is not possible. Only in specific sites called as the cloning sites, alien DNA has to be linked. So what is this alien DNA? It is nothing but the foreign DNA which we got from plant A. So that alien DNA, if it has to be linked, the vector needs to have very few cloning sites. Those cloning sites, at least if a single cloning site is there, then such vectors are mostly preferred. So most preferred is 1 or 2 cloning sites. Well, it's okay. But vectors which are mostly preferred are those having only a single cloning site. And that cloning site will be capable of recognizing the restriction enzyme. It should be capable of recognizing the restriction enzyme, because in that cloning site also the DNA has to get removed. Which DNA? The vector's DNA has to get removed, right? So that they will be placed out of the 2 DNA strand, 1 DNA strand has to be removed, so that they will be placed for the foreign DNA to come and join there or to go and stick there. So that is why a particular single cloning site should be there. Whatever cloning sites, how many number of cloning sites are there in the vector, that doesn't matter. We have to concentrate on that particular cloning site which is capable of allowing the restriction enzyme to go and bind there, so that the foreign DNA can go and bind there, making space for the foreign DNA to come and bind there. So the presence of more than one recognition sites within the vector will result in several fragments which will complicate the process of gene cloning. So that is why instead of having more number of cloning sites, it is always better to choose a bacterial vector that is having only one cloning site, and that too, that cloning site should be capable of having the restriction enzyme, or capable of allowing the restriction enzyme to go and bind there. That is the restriction endonuclease enzyme to go and bind there. After that, what happens? The ligation of alien DNA, that is the sticking of the alien DNA, is carried out. That is, from where did the alien DNA come? From the foreign DNA, right? That foreign DNA will go and bind here. Say for example, here they have given different restriction enzymes, HIND3, then CLAV1, ECHO-R1, PVU1, PST1. These are different restriction enzymes that are there. So any one restriction enzyme should go and recognize. Say for example, one restriction enzyme will be there in that particular site. That particular restriction enzyme will go and bind there. It will cut this is nothing but the DNA itself. Don't think what is this. This is nothing but the DNA. DNA of the bacteria, right? Which bacteria? Escherichia coli bacteria that we are using as a vector to transfer the DNA. So now here we have Escherichia coli bacteria. What is there in the Escherichia coli bacteria's DNA? There are different sequences present or sites present. What are the different sites? Sites such as the origin of replication site, the cloning site, then we have the selectable marker region, all that will be there. Now at present we spoke about the origin of replication, we understood what it is. We spoke about the selectable marker, we understood what it is. Now we are concentrating about the cloning site in that particular bacterial DNA. Now in that particular cloning site, always we need to prefer such bacteria having one cloning site. So for that one cloning site, any one of the restriction endonuclease enzyme can go and bind. So when the restriction endonuclease enzyme goes and binds, what it will do? In that DNA strand, it will cut one of the DNA strands, small portion it will cut, right? Now that place is empty. So to that place, the foreign DNA will be inserted. Now when the foreign DNA has to insert, the foreign DNA should ligate with the complementary strand there, right? So to ligate, next step is ligation. So the ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. So we'll explain it to you by taking an example so that it will be even more easier. So we can ligate a foreign DNA, say for example at the BAM-H1 site. So BAM-H1 site means that site which is capable of binding to the restriction endonuclease enzyme BAM-H1. So therefore it is called as the BAM-H1 site. So here BAM-H1 site, will consider this particular site. So the foreign DNA can go and ligate here. Why the foreign DNA will go and join here? Why? Because here is the cloning site which is capable of recognizing the restriction endonucleases BAM-H1. If it was capable of recognizing the restriction endonuclease HIND3, the foreign DNA would go and bind here. If it was capable of recognizing the ECO-R1 restriction enzyme, then also the foreign DNA would go and bind here. But here what am I taking the example of according to your textbook? I am taking the example of that restriction endonuclease enzyme that is capable of binding in this cloning site, this region. Therefore, the BAM-H1 restriction endonuclease enzyme will be recognized by this particular cloning site. It will go and bind there. Once it goes and binds there, it will cut the portion of the DNA and it will remove. Now this place is empty, right? So to this place the foreign DNA should come and join. That foreign DNA will come and join in this place now. So that particular process is called as ligation of the DNA. So now here what the DNA strands are there? DNA strands of the vector is there. One DNA strand of the vector is there. One DNA strand of the foreign DNA is there. So that is how it works here. So, site of tetracycline resistance gene in the vector PBR322. Here they have taken a particular vector, PBR322 vector is being considered. This is very important because they are usually asked this in the exam to draw the diagram of the particular vector PBR322. So, here this vector is having a selectable marker region which is resistant to the antibiotic tetracycline. So that particular region, now the foreign DNA has gone and incorporated. It has gone and bound there. Next what happens? The recombinant plasmid will lose the tetracycline resistance. Now the tetracycline resistance of the recombinant plasmid will be lost. Why will it lose the tetracycline resistance? Because here this portion of the DNA strand of the bacteria was removed and what was introduced? Foreign DNA was introduced, right? Foreign DNA is introduced. So that is why one more DNA came and got inserted here. Now will this gene show resistance to tetracycline? No. Why? Because bacteria DNA was removed, one strand of bacteria DNA was removed and in that place the foreign DNA was added. Therefore, the vector will now, the plasmid will now lose its resistance towards tetracycline antibiotic because of the insertion of foreign DNA. But even then it can be still selected out from the non-recombinant ones. Now, how can you remove when this bacteria, after introducing, what is the function of the selectable marker region? It is to remove the non-recombinants, right? Those during multiplication, it is not that all the clones will be there in all the bacteria once the bacteria multiplies. Say for example, I have taken a petri dish. I have added a bacteria here. Now what is there in this bacteria? In this bacteria, what is there? It is containing the plasmid. In that plasmid, what is there? It is having the foreign DNA. Now this bacteria will grow in the petri plate, right? Many numbers it will produce. Do you think that all the bacterial cells will have the foreign DNA? No. Those bacterial cells which are not having the foreign DNA, which are called as the non-transformants, that should be removed here. Now if it has to be removed, then this particular first plasmid, that is bacteria that is there, it should have the antibiotic resistant region, right? So which antibiotic resistant region was there? That region which was resistant to tetracycline. Now after the introduction of foreign DNA, that region is no longer resistant to tetracycline, or that particular bacteria lost its capacity to tetracycline. When it lost its capacity to tetracycline, how can it find out that which is the non-transformant? So in spite of that, we can find out which is the non-transformant. How we can find out? We can find out by plating the transformants on the tetracycline containing medium. Now what I will do is, from this I will take it out and I will plate it in a media containing tetracycline. So what is there in the media? Tetracycline, right? If the foreign DNA got introduced into the host, then it will grow well, or it will grow well in tetracycline media because it is not having any resistance gene there. If it is not introduced, it will not grow. So that is how we can find out. So the transformants growing on ampicillin containing media are then transferred on a media containing tetracycline. Recombinants will grow in ampicillin containing medium but not in the tetracycline medium. Again, what can be done after growing here? We'll put it into another media. Now in this media, which antibiotic is there? Ampicillin. Ampicillin, will it be recognized? It will be recognized. Why? Because in ampicillin, nothing went and bound there. No foreign DNA did not go and bind here. Foreign DNA was bound where? In this particular region which was resistant to tetracycline. But ampicillin is active, right? So therefore, we'll transfer it into ampicillin media. When it grows in the ampicillin media, those bacteria which are showing resistance towards ampicillin will grow. Those which are not showing resistance will not grow. So therefore, those which are not growing will be considered as the non-transformants. They will be eliminated and only the ones that are growing will be selected. Now, what are those selected ones? They are the vectors. Those vectors will be transferred into the plant. So that is how it is done. So this is a very vast topic and it is quite interesting if you understand the concept very well. So, but the non-recombinants will grow on the medium containing both the antibiotics, and in this case, one antibiotic resistance gene. It helps in selecting the transformants, whereas the other antibiotic resistance genes which will be inactivated. Why did it get inactivated? Due to the insertion of the foreign DNA or the alien DNA. That inactivated gene will be helpful in selecting the recombinant. So in an easy way, how it is done, I've already told you about it. So after selecting the recombinants, those will be transferred into the plant. So therefore, why will only those be transferred into the plant? Because they are the ones that will be having the foreign DNA. Our main aim is to introduce the foreign DNA into plant B, right? So that is why we will select only those having the recombinants and we will put it into plant B. So there also, when the bacteria multiply, the DNA will get incorporated in the plant B cells DNA as well. So all these are the same thing. So therefore, alternative selectable markers have also been developed based on the type of where the foreign DNA goes and binds, the selection of the transformants and the non-transformants will depend on. So that is why alternative selectable markers are also being identified. So therefore, here alternative selectable markers have been developed which differentiates the recombinant from non-recombinants. Apart from just ampicillin and tetracycline, other selectable markers have also been developed, but the most common ones used are tetracycline and ampicillin, and the same thing I've explained here as well. And in this, a recombinant DNA will be introduced within the coding sequence of an enzyme beta-galactosidase. And this beta-galactosidase results in the inactivation of the gene for the synthesis of the enzyme, which is referred to as the insertional inactivation. So, this is how it is done. So, next, in the presence of a chromogenic substrate, it will give blue colored colonies. What will happen there? Along with that, a substrate will be added which will give a blue color. So, wherever blue color patches are seen on the that is antibiotic media, those will be considered, those regions will be considered to be having bacteria carrying the foreign DNA or having the recombinants. Only those will be selected and the unstained ones will be eliminated. So, presence of insert results into insertional inactivation of beta-galactosidase gene, and the colonies will not produce any color. So, these are identified as recombinant colonies. So, these recombinant colonies will be selected and they are the ones which will be used as a vector and introduced into the plant cells. So, next is, what are the vectors for cloning genes in plants and animals? So, one of the best vector for cloning genes in plants that is used is Agrobacterium tumefaciens. So, Agrobacterium tumefaciens is actually a pathogen. This pathogen is capable of infecting a large number of dicot plants and it is capable of introducing its DNA. What it will do is, it will go to a plant, it will enter into the plant and it will insert its bacterial DNA inside the plant. That bacterial DNA will go and somewhere it will bind with the plant's DNA. So, when it binds with the plant's DNA, that particular DNA is called as tDNA. So, that tDNA is present in the Agrobacterium tumefaciens. So, that tDNA will go and incorporate into the plant's DNA and it will start to produce tumor cells. So, that is when you can see tumor growth in the plants because of the production of the tumor cells. So, similarly, there are some in animals, some retroviruses are used. So, these retroviruses have the ability to transform the normal cells into cancerous cells. So, these are also used for genetic recombinant technology. So, in plants, the most common vector that is used is Agrobacterium tumefaciens, and in the case of animals, the retroviruses are used. So, the tumor inducing TI plasmid, which is called as the TI plasmid of Agrobacterium tumefaciens. So, here what is selected? The TI plasmid is selected. So, how we saw the plasmid, that is a PBR 322 plasmid, right, of Escherichia coli, just like that here. The TI plasmid of Agrobacterium tumefaciens is selected and that will be modified as a cloning vector. Why it should be modified? Because when we introduce into the plant, it should not produce any disease, because Agrobacterium is capable of producing disease in plants, that is tumor growth in plants. So, that gene which is responsible for the formation of tumor will be inactivated or it will be suppressed. That is why it is called as modified. The TI plasmid should be modified before it is introduced into the plant. But it is capable of entering into the plant. So, that is why this plasmid is preferred. So, before introducing, we have to modify the TI plasmid. So, the modified TI plasmid will be modified into a cloning vector. Therefore, because it is modified. So, as I told you, because the gene that is responsible for producing the disease, that will be inactivated or that will be modified. So, that modified vector, if we introduce in, or plasmid, if we introduce into the plant, it will not be capable of producing any tumor. So, therefore, it will become no more pathogenic. It will lose all its pathogenic property and therefore, it will be introduced into the plant in order to deliver the gene of our interest. Similarly, in the case of animals also, retroviruses are used. So, in retroviruses also, it produces tumors, it produces cancer cells, right? But because it is capable of infecting the host very well, this is used as a plasmid. Here also the retroviruses should be modified in such a way that it is no longer capable of producing cancer cells when introduced into a animal host. So it will be disarmed, it will be made inactive, and then it will be used to deliver the desirable genes into the animal cells. So once a gene or DNA fragment has been ligated into a suitable vector, it will be transferred into a bacterial, plant or an animal host. There it will multiply, and when it multiplies, the desired foreign gene will also get replicated and it will get incorporated in the plant via animal body. Therefore, a hybrid plant or animal or a genetically modified plant or animal can be produced. So that is how it is done. And since DNA is a hydrophilic molecule, means it is water loving, right? It cannot pass through the cell membrane. And in order to force the bacteria to take up the plasmid, the bacterial cell must first be made competent to take up the DNA. So, how is it made competent to take up the DNA? So, DNA is hydrophilic in nature. It cannot, and it is hydrophilic, and the bacterial cell wall is, say for example, or the plant cell is made up of, or the bacterial cell maybe it might be made up of the lipid. So that lipid's head is hydrophobic, and this water loving DNA cannot pass through the hydrophobic lipid. So how can it be made to pass? So before going into incorporating the DNA into the bacterial cell, first we need to make the bacteria capable of accepting or allowing the DNA, foreign DNA to enter into its cell. So this is done by treating them with a specific concentration of divalent cation such as calcium. So first the bacteria that is selected as a vector will be treated with calcium. So once it is treated with calcium, it will be capable of allowing the foreign DNA to enter into its cell. So that is how it done. Therefore, the pores will be formed on the cell wall of the bacteria when it is treated with calcium, and through those pores, these particular foreign DNA can enter into and get incorporated in the plasmid of the bacteria. So that is how it is done. So recombinant DNA then can be forced into such cells by incubating the cells with recombinant DNA on ice. So first what they will do? So first what they will do is they'll take the bacteria. Those bacteria, say for example Echo R1 bacteria, they will take. Now the Echo R1 bacteria is a vector, right? It has a plasmid. Say for example it has PBr322 plasmid. Okay. So this Echo R1 bacteria or vector, the foreign DNA has to be introduced. Now the foreign DNA, will it enter? It will not enter. Why will it not enter? Because the foreign DNA is hydrophilic in nature. It cannot pass through the hydrophobic wall of the bacteria. Therefore, first the bacteria that is there, it will be treated with calcium. When it gets treated with calcium, pores will be formed on the wall of the bacteria. Therefore, when the foreign DNA is introduced into the bacteria, the foreign DNA can pass through the pores or the holes and it will get incorporated there. So now it has to go and fix itself or ligate itself with the bacterial DNA. How will it happen? So for that particular process, the bacteria has to be incubated. So the bacterial cells will be incubated along with the recombinant DNA on ice, and it will be, after putting them in ice, a heat shock will be given. That is, it will be put in a temperature of 42 degrees Celsius. So, and again it will be put in ice. So when it is first in ice, then under high temperature, and then again back in ice. So this change in temperature will enable the bacteria to take up the recombinant DNA. So that is how the recombinant DNA is introduced into the bacteria. And once the bacteria takes up the recombinant DNA with the help of the restriction enzyme, previously how I explained, like that the DNA will go and incorporate itself in the particular site. So that is how it will happen here. And there are other methods also which are followed apart from treating it in ice and or putting the bacteria in calcium, treating it in ice and in hot ice hot, ice like that, treating it in different temperature. Instead of that, there are also other methods how we can make the DNA to enter into the bacteria. Three important methods are there. One is micro-injection. These two are very important, ballistic or gene gun method. Micro-injection. Here, small injections are used, that is micro-injections. To that micro-injection, the recombinant DNA will be put and it will be directly injected into the bacteria. So, this is about the micro-injection. As you can see here, using the micro-injection needle, so here the foreign DNA will be there, and using this micro injection, it will be directly introduced into the cytoplasm of the bacteria or the host cell. So that is how it is done, or into the nucleus of the animal cell. So next is ballistic or gene gun method. Here this ballistic or gene gun method is usually suitable for plant species, and here the cells are bombarded with high velocity microparticles of gold or tungsten. So here a gun-like structure will be used. Inside this gun-like structure, DNA gold will be used. To that gold particle, the DNA will go and bind, and it's just like a bullet. How the bullet will get fired very fast, right? Just like that, the gold particle will act like a bullet. What is surrounding the gold particle? The DNA of our interest is there. So that will be shot into a plant or a animal species, and that is how the DNA with the gold particle which is acting like a bullet, it will go and enter into the plant or the animal species. So that is how it is done in ballistic or gene gun method. Next is disarmed pathogen vectors. So in disarmed pathogen vectors, what they do is, it will allow the pathogen to infect the cell, like how we use Agrobacterium tumefaciens and retroviruses in animals, right? That is disarmed pathogen method. So there again, how it is done, I have told you. Like they'll make them non-pathogenic, and then they will insert it into the plant or allow them to infect the plant or the animal cell respectively, and therefore once it infects and enters into the foreign DNA, obviously will enter into the plant or the animal cell. So that is how it is done in the case of disarmed pathogen vector. So these are the three methods which can be followed other than the previous method that I told you. So this is how the bacterial DNA can be, foreign DNA can be introduced into the host cell. So previous method is used to, don't get confused, previous method wherein heat shock and cold shock is given, that method is used to introduce the DNA into the vector or into the bacterial cell. And after it is introduced into the bacterial cell, it has to be introduced into the host organism. For that, microinjection, ballistic or gene gun method and disarmed pathogen vector method is followed. So this was about this particular session wherein we studied about the exactly how the cloning vectors are used and how the cloning vectors can be introduced into the host cell. So I hope you understood this session well. In the coming session, let us discuss about the process of recombinant DNA technology. We'll continue with the process of recombinant DNA technology, and also we'll find out at the end how we can obtain the foreign gene product, because the main aim of biotechnology is to obtain a foreign gene product, right? So how it is obtained we shall look into. So we shall meet again in the coming session with these two topics. Thank you.